Assistant Professor in the Department of Horticulture at Michigan State University, and welcome to Plants in Python. So in today's lesson, I'm going to provide an introduction to plant genomics and plant genome assembly. And so you could spend a whole course talking about plant genome and plant genome assembly, but today I just want to provide a brief, roughly half an hour introduction to the principles of genomics and genome assembly. And so the plant biology-based learning objectives of today's lesson are to understand what makes up the genome and how genomes vary between plant species. We will discuss the current state of plant genomics, talk about what's been sequenced and how, provide a history and some of the fundamentals of DNA sequencing technology and how this has changed over time, and also go over some of the principles of plant genome assembly. And in later lessons in this notebook, we will be talking about BioPython, which is a workhorse in Python for manipulating and working with DNA sequences, and then we'll actually be running genome assemblies on HPCC using some of the principles that we talk about today. Okay, so first, fundamentally, what is the genome? And so the genome is what makes up all the genetic material within a cell. So this could be anything living, plants, animals, fungi, pick your favorite species. And so the genome at its core contains the things we care about most, which are the genes. And so these are the things that get turned into protein and that function in various cellular processes. But genomes are also filled with repetitive elements. And so these are typically non-functional, but they do have some function and they make up the majority of plant genomes. And then we also have various intergenic sequences. These are sequences between genes that can have regulatory functions. They can have structural components, all sorts of things. And so what I'm showing here is a karyotype, and so this is kind of a splay of all of the chromosomes in a living organism, and so the genome is contained within chromosomes. And so on top here, this is maize or corn chromosomes, and then this is what the human chromosomes look like. And so just for kind of a reference point, the human genome and the maize genome are pretty similar in size, and so 3 gigabases or 3 billion bases versus 2.3 gigabases or 2.3 billion bases. And they contain different elements. And so we sort of use different terminology when we talk about genomics. And so there are 3 abbreviations, KB, MB, and GB. And so 1 KB equals 1,000 base pairs of DNA. So this is 1,000 A's, T's, C's, or G's, the fundamental components of DNA. A megabase, or MB, is 1 million bases of DNA, and gigabase is 1 billion bases. And so if we're talking about the maize genome, we could say 2.3 gigabases, 2,300 megabases, or much larger in kilobases. And within a plant genome, about 1 to 20% of the plant genome is coding. So this makes up the genes and gene sequences. The rest is intergenic or classified as other. And so some regions of the genome are functional, so they have structural or regulatory functions, and others have unknown or no known function. And so in the case of the human genome, this is probably the most well-characterized one. There have been decades of effort with projects like the ENCODE project to identify what all the sequences in the genome are doing. But at the end of the day, we still don't know what a lot of parts of the genome are doing, and a lot of them also have no function. And in general, plant genomes have on the order of 20,000 to 200,000 genes, and this depends on the species, it depends on the polyploidy, and a couple other elements. And so plant genomes vary massively in size and content, and so generally plants have about a 10-fold variation in gene content. So from about 20,000 to about 200,000, but they have over a 2,400-fold variation in genome size. And so here, we're showing down here, this is at the smaller end, so things like diploid strawberry, Arabidopsis, the sort of model workhorse plant, and those would range from on the order of 100 to 250 megabases, all the way to Paris japonica, which has the largest known plant genome size of about 150 gigabases. So this is, for reference, about 50 times larger than the human genome. And a lot of this is driven by repetitive elements, or these sequences that originated from viruses that have 99% identity to each other, and they just keep proliferating and making the genome larger and larger. And so as you can imagine, this provides a lot of challenges when we're thinking about genomics and how to assemble genomes from different species. And so in general, most crop genomes have been sequenced. So to date, and this is in 2021, but this will probably be out of date relatively quickly, over 1,500 plant genomes have been sequenced and released. And so this includes most crop species, many wild species, and model ecological species. And there are thousands more in various stages of completion. And so it used to be quite expensive. And so the human genome, for instance, costs an estimated $4 billion, which is an incredible amount of money and effort from dozens of institutions across the world. And the rice genome, which was one of the first plant genomes to be sequenced, cost about $100 million. And nowadays you can assemble a high quality, essentially complete genome on the order of $1,000 to $5,000, depending on the sequencing technology you're using and the size. So it's been really a stark contrast in the way things used to be. And so here's a plot just showing sort of the submission date and the quality, or Contig N50 of genomes. And so this starts in 2008, and you can see in general over time the quality has gone up exponentially. So this is a logarithmic scale, and the number of sequences that have been published has also gone up exponentially each year. And so now it's much cheaper and much easier to assemble reference genomes. Over the past 20 years, plant genomics has been dominated by what we refer to as the global north. And so the vast majority of plant genomes have come from sort of 3 regions throughout the world. So we have sort of the United States and Canada, various countries in Europe, and then mostly China and Japan and South Korea. And so there have been comparatively very few reference genomes that have come out from the global south. And so this would be Central and South America, Africa, and then parts of Southeast Asia. And so this is a massive problem because the majority of crop species were domesticated in sort of these tropical regions. And a lot of the natural diversity is still found in these countries, and a lot of these wild species are also found there. And so there are inequalities in plant genomics, and this is something that's changed over the past couple years as the cost has gone down, but there is a lot of work that still needs to be done of emphasizing sequencing and community resource building in these various parts of the world. Okay, so going back in time a little bit, the first plant genomes, the first genes in general were sequenced using Sanger sequencing technology. And so this was the first sequencing technology that was developed. And we won't go into too many details about how this was done. Again, you could spend hours talking about this. But in essence, it produced very high quality, very low error rate sequences that were on the order of 500 to 1,000 base pairs. And so this technology is PCR based. And so what would happen is you would have your DNA sequence of interest. You would be amplifying it with some of these terminal nucleotides, and then when they get incorporated, that stops the amplification of that DNA sequence. And so this used to be done with radioactive markers. And so what you would do is run PCR for 30 to 40 cycles. It would randomly terminate at different positions in the genome, or in the sequence of interest that you were sequencing, and then you would run them on these very long polyacrylamide gels, and then look at the sequence composition from there. And so here, for instance, at the very top, this, actually let's start at the very bottom here. So this sequence would be the first base pair, the shortest base pair would be a T, followed by a G, followed by what looks like two T's, and then an A, a C, another A, etc. And so this was the olden days, and so these gels would be on the order of meters long. It's kind of dangerous working with polyacrylamide, and so a PhD thesis in the late 80s, early 90s could be running a couple dozen of these gels and sequencing a single gene or several genes. And then the subsequent technology used these different fluorofluors, and so we would incorporate these chain terminating fluorescent nucleotides, run them out on capillary electrophoresis, and then this fluorescent label would correlate with the different sequences. And so in this case, you would have the first base pair would be a G, the second would be a G, CCA, CCCT, etc., etc. And so this was very labor intensive, and so you could only really run 96 samples in a 1 to 3 hour block or a single plate, and it was very costly. And so you can still use Sanger sequencing nowadays, but the cost is $1 or a couple dollars per sample, and it's very low throughput. And so for the first genome sequencing projects, so things like the human genome, the Arabidopsis genome, the yeast genome, etc., we had these massive sequencing centers where you would have each one of these machines would have 96 well plates or 384 well plates. It was sequencing small pieces of fragments of DNA at a time, and you'd have hundreds of these ABI Sanger sequencers that would be running around the clock, assembling sequencing small fragments. And so in these massive sequencing centers, floors of buildings, hundreds or thousands of these individual sequencers running at a time, and lab technicians coming in and replacing the samples, and it was a very slow iterative process. And this is why the human genome costs $4 billion to sequence, and why individual institutions or even countries could only sequence one chromosome or one chromosome arm at a time. So this was a very expensive and very slow process. And so after that, we had the second generation sequencing technologies, which we're going to be working with some of this data in this course, but I won't get into that. Second or next generation sequencing. And then what I want to jump to is this third generation sequencing technology. And so this is what we have now for the majority of genome assemblies, and it's kind of the workhorse of plant genomics. And so this is a fundamentally different sequencing technology than the first or second generations. And so in those cases, they relied on a PCR reaction to amplify a single piece of DNA into millions or billions of sequences that we could then analyze and identify at the single base pair. But instead, these sequencing technologies rely on running a single molecule of DNA through a pore and either running a DNA polymerase enzyme, or basically DNA synthesis, as the case in PacBio, or taking a single DNA molecule, unwinding it, and looking at the disruption of current as we see in Nanopore sequencing technologies. And so the benefit here is we're able to get these massive fragments of DNA up to several hundred kb or several 100,000 base pairs, and we're looking at what's happening in real time in a single molecule. And so the way this sequencing technology works is we have a single DNA polymerase enzyme, and so polymerase is the enzyme that basically copies our DNA. We have each of the nucleotides here that are phospholinked to a different fluorofluor that fluoresces at a different color for the G's, A's, T's, and C's. And then what happens is as our DNA molecule feeds into here, the DNA polymerase will incorporate a different nucleotide, and then when it gets incorporated, it will remove this phospholink tag, and then that will create a fluorescent pulse. And we can record that fluorescent pulse as it will correlate with a different nucleotide. So here I have this nice animation that shows this process occurring in real time. And so here is, again, this DNA polymerase enzyme. Here is our strand of DNA. And each time it incorporates a base pair, you can see this fluorescence that's given off. And each one of these, the nucleotides, A's, T's, C's, and G's, have a different fluorescence. And then it gives off that signal. A laser records that signal, and then you can identify what the nucleotide is from that. And so this is a really powerful sequencing technology. We're doing this in real time, And so over a period of a couple minutes to a couple hours, you can sequence this massive fragment of DNA that can be a couple 100,000 base pairs at a time. And so this is what it looks like in real time. And so this is an actual sequencing cell for PacBio. So there are hundreds of thousands to millions of these individual pores. And you can see each time this pore is incorporating nucleotides. So here is this red one. So that would be, I think, a G. And then we have some greens, some blues, and some yellows. And so you have hundreds of thousands of these pores going at a time, each one working on a single DNA molecule. And so at the end of the day, you can get millions of these sequences in a period of about 24 to 36 hours that have really long lengths. And so it's really long, really high quality data. Now, the only problem here is that since we're incorporating this in real time, so you're doing single base pair at a time, it is somewhat error prone. And so we're recording a single nucleotide being incorporated. And so the error rate of these reads is on the order of 1 to 10% errors. And so that's kind of a problem. And so the most recent iteration of this technology from PacBio is called PacBio HiFi reads, or high fidelity reads. And so instead of a single circular or single linear piece of DNA going through a polymerase once and us having some errors in here, because again we're recording a single base pair being incorporated at a time, what happens is you have this long strand of DNA that is circularized. So it's turned into a circle here, and then this DNA goes through the polymerase. Instead of it going through once, as you would have for a long linear piece of DNA, it goes through several times. And by going through this same circle several times, we sequence each base pair in that sequence maybe 3 to 20 times, and then you can find a consensus from that. And at the end of the day, we get this very high quality reads that have an accuracy of 99.9+%. And so instead of having it go through once, where the error rate is maybe 1 to 10%, we go through it as a circle a couple times, and then we get it down to these very high, very accurate reads. And so this is really important in the next step of genome assembly. So we'll come back to this. So the other big sequencing technology that's on the market now for third generation is from Oxford Nanopore. And so this works with some similar principles, but also some differences. And so again, we're looking at sort of the single molecule process, and it's occurring in real time. And so here we have a pore that's embedded in a membrane, and then we have another enzyme here, so it's basically like a helicase. And what's happening here is instead of our DNA being run single strand through here and synthesized, it's running through this pore, and then we can look at the difference of current. So the A's, T's, C's, and G's have different base pair compositions, and so they'll cause a difference. If we run an electrical current through this, they'll cause a different electrical current. And what you can do is run these DNA molecules through this, and then look at the patterns of electrical currents, and then use machine learning to identify what's causing that difference. So here you can see the different sequences are causing these different waves, and then we can from that figure out the sequence of A's, T's, C's, and G's. And so this is somewhat similar in that we produce this very high quality DNA, run it through this machine, and then we're getting these single molecule sequences. And so what's great about this one is the device itself is quite small, so you can plug it into a USB port, and you can take it anywhere. But again, the downside to this, very similar to PacBio, is we're looking at a single DNA molecule being pushed through a pore, and in this case we're looking at very subtle changes in ionic differences and electrical signals, and so this sequence is also error prone. So again, it's on the order of 1 to 10%. And so we can correct for this, but it makes some challenges downstream in genome assembly. And so in general, from both of these sequence technologies, we get very long reads. So on the order of 50,000 base pairs, or 50 kb, all the way up to 250 or 300,000 base pairs, and you can even get higher than this. And in general, the limitation now is really no longer the read technology, but the quality of the high molecular weight DNA that you put into here. And so in general, if you remember from Sanger sequencing, we're looking at 500 to 1,000 base pairs. Here we're looking at individual reads that are 50 to 200,000 base pairs. So this makes it much easier to do genome assembly. And so you can do some of this in the field. And so this is an example. This was from the Zika virus outbreak a few years ago. And here we have this Nanopore sequencer that's out in the field. You can quickly isolate some DNA, run the sample through the Nanopore sequencer, and then you can look for your virus of interest. So maybe this was Zika in this case. People have used this for COVID. They brought these to Antarctica and sequenced soil microbes and things. So you can really do the science anywhere. All that requires is some simple molecular biology tools or molecular biology equipment to isolate genomic DNA, and you can sequence things anywhere. So this is really powerful sequencing technology, and we're kind of continually moving in this smaller, quicker, easier direction. Okay, so now we've talked a little bit about the various sequencing technologies. Now we're going to talk about how we put it all together. And so when we isolate DNA, so we have this chromatin in a plant cell, so this is just the kind of chromosomes that are spread out like spaghetti that make up the genome. We have broken this up into random pieces, and then we've sequenced these large chunks of DNA using maybe Nanopore or PacBio based sequencing. And so the next goal is to take these random chunks of DNA and assemble them back into chromosomes so that we can identify genes, regulatory elements, and other sequences of interest. And so the question is, how do we do that? And so we're going to talk a little bit about the fundamentals of genome assembly and kind of where we go from having a genome assembly to some of the downstream analyses we can do. And so the way that we generally do genome assembly is looking for overlaps between sequences. And so I have a couple examples here that are based on text overlaps to kind of understand the concept, and then we'll talk about how DNA overlaps work. And so perhaps we have an example here where we could consider each one of these partial sentences as a read of DNA. And so here we have, the first one here is my car broke down in the deep. The second one is woods, period, suddenly I heard a. And then the third one is in the deep, dark woods, period, suddenly. Right, so these three should go together in some way, and so we can look for overlaps between these and assemble them into a longer fragment of text. And so if we look for overlaps here, we have this first read, or this first part here, which is my car broke down in the deep. And then in the deep aligns with this second fragment here, in the deep dark woods. And then this second fragment aligns well with this third fragment. So if we put these three together, we get this consensus sequence of my car broke down in the deep dark woods, period, suddenly I heard a blank, and who knows what that is. But in this case, this is a very simple sequence, or in this case a sentence, or a sentence and a half, where we have no repeats. It's very clear how the order goes, and there's no ambiguity. Plant genomes especially, but genomes in general, are much more complicated than this. So what happens if we don't have a perfect match? And so here we have an example of these 5 sentence fragments here, talking about a story about a farmer and various things that that farmer would do. And so we can take these 5 fragments and try to figure out the order in which they go. And if we kind of were to cut these out on paper and then try to organize them, in the beginning, we would have the dog walked through the corn, and then we have this nice read here where we can overlap the corn together, and then the dog walked through the corn with the farmer. But then once we get to the farmer, we don't know what the order is, right? So it could be the farmer with the apples on the farm harvested the corn after, or the farm baked a pie with the apples. And so this is an example where, given this limitation, we don't know what the order is, right? Does the farmer correspond to this sentence, or this sentence? And so this is a major issue in genome assembly, and we encounter this problem a lot. And so here I'm showing an example where there are kind of these 3 overlaps or 3 repeats that are kind of difficult to parse out. But in reality, genomes can have hundreds of thousands of very similar sequences. And so we have to apply some really complex algorithms to try to figure out the order of how these things go together and what they mean. And so this is sort of the correct order of this, and so I would usually do this as an activity where we print out various chunks, And you can see here there's a lot of ambiguity here. So if you were to try to read this right now with just these sentences as they are, it would be a lot of run-ons, and it's hard to figure out where sentences break or where they end. And so this is sort of like an analogy to a raw genome sequence. So let's say we have a whole chromosome. It's just a string of A's, T's, C's, and G's, and we don't know how to make sense of it. And so the next step would be annotation. And so in this case, if we apply periods to the end of sentences and we capitalize other sentences, it makes it a lot clearer what the story is here. And so this is kind of analogous to annotating a genome. And so in this first lesson, we will talk about how to assemble fragments of DNA together. And then we will talk in the next lecture about how to assemble sequences. And so how to go from this string of A's, T's, C's, and G's to identifying putative genes and sequences within the genome that are of interest to us. Okay, so for genome assembly, there are a couple different ways that we can do this. And so the most common is this sort of overlap layout consensus approach. And so this is very similar to the example I just showed, where we would align all of these sequence reads based on overlaps. And so here we can see that all of these reads here have this TTA here, and that's the simplest, and they overlap based on all of these reads, and we can take these individual reads, align them together, and then create this consensus sequence. So as you can see, if you were trying to do this as a puzzle, it would be way more difficult than if it had sentences in a book. And so the general sort of approach that these algorithms take is that we take these long reads, we align them together to correct any errors, and then produce these sort of long seed consensus reads. And then we take these pre-assembled longer reads, align them together, and then make a genome sequence from them. And so again, this is just based on overlap. So this is very computationally intensive because you are comparing basically every sequence against every other sequence and seeing exactly how they overlap. But at the end of the day, we get these really nice long fragments that then can be stacked together to make a consensus sequence for a reference genome. And we use some terminology when we talk about genome coverage. And so if we were to just sequence each base pair of a genome on average of once, we would be missing a lot of sequences, right? Because we're just sequencing random pieces of DNA, and so you need to sequence tons of DNA to make sure that you're getting every base pair in the genome. And so there's a term that we use called coverage here. And again, we have this consensus sequence down here, and then we can imagine that each one of these are read aligning to that. And so when we talk about coverage, it basically means the average amount that each base pair in a genome is sequenced. And so here we can count, this is a total of 177 nucleotides for this 35 base pair fragment. And so this would be an average of 7x coverage. So if we look at any given position, let's say this T here, we have it represented 1, 2, 3, 4, 5, 6, 7 times. So in general, each base pair would be sequenced an average of 7 times. And so if you could imagine we only had 2 or 3 times coverage, there might be some mistakes introduced here, there might be some gaps. And so generally in genome assembly, we're looking for an average coverage of 20 to 100x, where each base pair would be sequenced 20 to 100 times. And that allows us to make sure we've sequenced every part of the genome and that we're able to correct any errors or identify any differences that might exist. So overlap-based assembly approaches are really great because we're looking at these long-range overlaps between individual reads, and so we end up getting this very high-quality assembly from that. But the challenge is that's very computationally intensive. And so for overlap-based approaches, we're looking at how every single read in the assembly overlaps with all the other reads, and so that takes a lot of computational resources, and it scales exponentially for the amount of data that we have in the assembly. So we can't really do that for very large genomes. And so the other approach that we can use is to break up these reads into much smaller pieces that are computationally simpler to assemble, and we can use a de Bruijn graph-based assembly approach. And so here's an example with this fragment of DNA here. So this is a small fragment, but it's represented here as a circle So the circle starts with ATG and then it ends with GCA. Let's say that we assembled or sequenced this using short-read sequencing technologies. And so here, each one of these different colored fragments would represent a single read. And this is just looking at sort of their overlap. And so we can see here, this read overlaps with this read at these positions here, and so forth, to create this consensus sequence, or in this case, what we're referring to as a genome here. And so we can display this using this overlap-based approach here, or we can display it as a graph. And so this is displaying it as a graph. So here we have the first read here in red, and we can see that this read overlaps with this blue read here. So this is position 1. This blue read overlaps with this yellow read here. This yellow read overlaps with this green read. This green read overlaps with this brown read. And because this is a circle, the brown one also overlaps with the red one. And so this is a nice example of like a graph-based assembly, where from this we can see how these sequences overlap with each other. And so in a real genome, this is going to be much more complicated because we have millions to billions of sequences, but this kind of gives you a rough sense of how we can take this assembly and look at it in a graph-based approach. And so when we're doing a De Bruijn graph-based assembly, what we would first do is, instead of looking at the overlap of these entire reads, we can break them down into shorter sequences, such as k-mers. And so here, this is an example of a k-mer with a length of 3. And so here we could take this, basically this consensus sequence here, we could break each of them up into 3 base pair reads with moving over 1 base pair at a time, and then sort of creating this consensus sequence. And we can look to see how these k-mers overlap with each other based on, in this case, where the vertices are the k-mers and the edges are the pairwise alignment. And so in this case, we can see the first sort of k-mer would be this ATG, and then for moving over a base pair would be TGG, over one more base pair, GGC, etc., etc., etc. And then you can also see how sometimes there are these other sort of spurious overlaps. And so we can sort of create this cycle here that has all the possible combinations in which these sequences can overlap with each other. And then we can also just look at simply the ends of the sequences. These are the edges, and we can find a perfect overlap. And so here we have, starting again with this AT, we have the k-mer here of ATG. It's overlapping these past 2 base pairs here. This TGG overlapping here, etc. So we can go 1, 2, 3, 4, 5 over here to 6, 7, 8, 9, 10. And then we can reconstruct this sequence from this. And so the nice thing is we're breaking down these individual reads into all the possible k-mer combinations of a set length. And so in here we're using a k of 3, and then overlap of k minus 1 or 2. But in reality, we could have k-mers of 55 or 1055, and we can look at how those sequences overlap with each other. And this is computationally much simpler and requires a lot less resources than doing every possible overlap between every read that we have. And so there are a lot of different genome assembly programs. There are dozens that are currently used in the community, and a lot of these use several core algorithms that were developed that are computationally efficient and have been shown to work really well, but they have different downstream steps or different data processing steps. And so different programs were built and tested with different types of data. So some work really well using different sequencing technologies. So there are some that work well with older sequencing technologies, such as Illumina, which is a next generation sequencing, or Sanger sequencing, and others work well with these longer single molecule based approaches like PacBio or Nanopore. And they also work differently with assembling genomes with different characteristics. So there are some programs that are really good at assembling large repetitive plant genomes or animal genomes, some that work really well at assembling heterozygous genomes. So that's where the A and B copies of the genome would be very different from each other. And there are some that work really well at assembling polyploid genomes, where you'd have more than 2 copies of every chromosome in the genome. And so if you're running a genome assembly project, you can test different programs and find one that works really well for your project. And so here are a couple of the common ones that we use in 2021. These are probably the 5 workhorses that people use most often. But if you were to come back a year or 5 years from now, you probably would find a different series of programs. And each one of these is slightly better suited for genomes with different complexities and also different sequencing technologies. And so Canu is probably one of the more common ones. This was developed for noisy PacBio or Nanopore reads that had an error rate of 1 to 5%. But then there's this other program, HiCanu, that is kind of tweaked for working with these very high quality HiFi reads that we talked about that have an average error rate of less than 0.1%. And so programs are constantly being updated. But in this course, we will be talking and working with these last 2 programs here. And so to date, we think of genomes as being done, and the truth is the majority of them are not yet complete. And so this is an example of what the human genome looked like in 2019, although it has since been completed. And here, with this assembly graph, we represent simple assembled sequences as kind of these clean pieces of spaghetti here. And so we would refer to this as a simple contig, where this 800,000 base pair piece of DNA was assembled completely with no problems. There's no ambiguities here. So this is what we would refer to as like a nice high quality sequence. And then we have what we refer to as a heterozygous bubble here. So in this assembled contig, we have this single piece of DNA here that's assembled correctly. And then when it gets to this point in the fragment, it doesn't know if it should match this 80 kb region here or this 80 kb region here. And so this could represent a heterozygous bubble where in the genome, this chunk of DNA has very low heterozygosity, so the differences between the copy from mom and dad are very minimal. And then here there are a lot of differences. And so you kind of get these forks here where it doesn't know if it should go to this fork or this fork. In reality, it's much more complicated than this. So this kind of pile of spaghetti where you have like these sort of chunks here where these reads can align in lots of different ways, where it's sort of a hairball where it doesn't know if, you know, a contig here aligns to one of 100 contigs within this chunk here. And then you do see some large sort of simple fragments. This is what the human genome looked like in 2019. It's not 20 or so pairs of chromosomes. It's still thousands of different pieces. So here we're kind of blowing up one of these complex repeat regions. And so again, each one of these individual lines here represents a simple assembled contig. And so we can see like this blue one here, this green one here, this green one here, etc. And then each one of these connections here represents an ambiguity. So let's start with something simple, such as the connection between this green line. So in the assembly algorithm doesn't know if this edge of this green contig aligns to this purple one, or if it aligns to this blue one here. And so we have these sort of simpler ones where it's 1 or 2 ambiguities. We have cases like this where it might be a dozen ambiguities, where it doesn't know, for instance, if this piece here aligns to this piece, this piece, this piece, or that piece. And then we can get really complicated hairballs here where we have dozens or hundreds of fragments that all kind of interconnect with each other. And so this could be a repeat, like a transposon that might have a couple hundred copies in the genome, and so it doesn't know which copy corresponds to which chromosome. These can be highly complex repeats, such as subtelomeric or telomeric or centromeric sequences, where it's these long tandem repeats of the same unit, so it doesn't know where they go together, or it could be other complex regions in the genome. And so these are still major challenges for genome assembly. But the human genome was essentially finished earlier this year in May 2021. And so here, instead of having these chunks of spaghetti, we have each one of the human chromosomes here represented by a much simpler assembly graph. And so this was aided by these long, high-quality HiFi reads tons of data and some computational algorithms that will improve the assembly where there are only a couple ambiguities left. So like, say this chunk of chromosome 6, where it could be these ribosomal DNA copies that can't quite figure it out, but we're getting to the point now where the sequencing technologies, the quality, and the read length allow it so that we can get through any repetitive or any difficult region and assemble them into complete pieces. So that concludes this lesson on plant genomics and plant genome assembly. Again, I just tried to provide a brief overview of these different areas, but if you're interested, you could take a whole course on this discipline. But hopefully you learned the following today. Learned about what makes up the genome and how these genomes vary between species. And so in the next notebook, we will be doing some comparative genomics where we look at what's in the genome and how that varies more specifically between species. We talked about the current state of plant genomics and how thousands of genomes have been sequenced, and this has been aided by improvements in sequencing technologies. We talked a little bit about the history and the fundamentals of DNA sequencing technologies and how this has changed over time. And then we talked about the principles of genome assembly, how fundamental algorithms work, and the different programs that we can use to do this. And so in the remainder of this notebook, we will be talking about BioPython, which is kind of the workhorse of working with DNA sequences in Python. And then we'll also be running some genome assemblies of our own using real data. And then in the very last activity, we will be using BLAST to identify the genome that you sequenced.